Hey everybody, happy Wednesday. Okay, give me just 1 sec. For some reason, my computer is trying to download a new version of Google Chrome. I don't know why. And okay, it went away. Perfect. That's what we needed. Okay. Don't you hate that when your computer's just like, hey, I'm going to download this random app that you don't actually need right now? And I'm like, actually, I have things to do, but I appreciate your service at this time. So hopefully I'm not the only one who has that. But woo, I fixed it. So woo, it's Wednesday. I'm back. I'm Jessica Nadsam. I've done several streams for Fiveable, and I'm so excited to be doing another one. So here we go. So woohoo. Y'all want to talk about some feedback mechanisms? Because that's what we're going to do today. We're also going to talk about the cell cycle and its regulation, because that's going to be really important to maintaining your health and well-being. I don't know about y'all, but I like living, and I rely on my cell cycle to copy all my cells properly in order to do that. I also rely on my feedback systems to maintain homeostasis, which is my basic zen that keeps me not dead. So we're going to talk a lot about those and how those work in your body today, but you're probably ready for the stuff that's going to be like on your tests in the future, so we should probably go ahead and start talking about that. So I'm going to go ahead and share my screen. And we're going to look at these slides, and it's going to be great. La, 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 la. Please ignore my singing. I'm really sorry. I'm really hyper. It's Wednesday, but it feels like Monday, and I have a lot of energy apparently. So I apologize that y'all have to put up with this. But on the bright side, hopefully it will be an engaging experience for those of you who wish to understand feedback mechanisms and the cell cycle. So whoop, whoop. So it is Wednesday, January 29th. It is 8 p.m. Eastern time, and we're going to talk about this stuff. I have already taught this stuff in my classes, so this is a bit of a redo for me, and I'll be honest with y'all. I've taught this 4 times in the past, week, so I'm a little bit like, okay, let's get this done. But I also really want you guys to understand it, not only for the purpose of the AP exam, which is May 11th of 2020 at 8 a.m., but also because I truly believe it's important to understand how our bodies work and how they keep us balanced in homeostasis. And both of these processes are so critical to literally keeping you alive. So let's talk about how they work. I'm going to go ahead and make this full screen for you because you don't want to see all my notes and stuff. That's not relevant to you. What is relevant to you guys is these slides. So whoop, whoop. As I said a few minutes ago, I am Jessica Nadsam. I am a 5th year AP biology teacher in a teeny tiny town in Arkansas. We do not ride our horses to school, in case that's the first question you had. If that's the first question you had, I need you to expand your horizons and the TV you watch. So let's take a look here at what this all actually means. So quick plug, if you don't already, you should definitely follow us on Twitter, Instagram, YouTube, Think Fiveable. We have lots of great content that we want to share with you. I don't know if you've been in the loop about some of our changes, but over the new year, our awesome founders have started looking at even more ways to get you guys resources to help you with your AP classes. We've got study guides, blog posts, videos are more differentiated to what you guys actually need, trivia, practice. So we think we're doing some pretty cool stuff. So we would definitely appreciate if you would take a look at all of it and stuff. I personally have written some of the content, so obviously I'm a little bit biased. But as someone who's been writing study materials for high schoolers since 2015, I feel like my bias is well justified. This is good stuff. You should definitely look into it. And in the meantime, here's another great thing I've written. It is the AP Biology review of feedback mechanisms and the cell cycle. Whoop, whoop. As usual, I made sure to get lots of GIFs because let's get real. We're all just here for things that are shiny and blink a lot and animated. I can't read words on a slide. I get bored teaching words on a slide. So I have to have all this stuff. I hope it works for you. If your learning style is more verbal, congrats. I'm going to talk for an hour straight. If your learning style is more kinesthetic, try writing this down as you go. I'm a big kinesthetic learner. If I don't write something down, it doesn't stay in my brain. So hope that works for you. If not, if you're visual, I have lots of memes for you. If you're more verbal, I shall talk the most of the time for you. So here we go. You've already heard my little spiel, my introduction about who I am, Jessica Nadsum, whoop whoop. We're going to talk about feedback. We're going to talk about the cell cycle. We're going to talk about cell cycle regulation. So as you can see here, I've got this little dude who's just saying, go, 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 start, 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 stop, 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 stop. Feedback cycles are all about getting more, or stopping a process and making it go back to whatever the original was. We're going to talk about both of those types today. We're also going to talk about the cell cycle. So when you start out with one cell and it splits, and then you have two cells, whoop, whoop, which you do like all day, air day. It's really important. And then we're also going to talk about cell cycle regulation. So how do we make sure that our cells divide properly? And what happens if they don't divide properly? Spoiler alert, we really just want them to divide properly. So let's go ahead and get into these. Before I get started, I see Madison has already said hello. Hello, Madison. So nice to see you. My name is Jessica Nadsum, as I've introduced myself about 12 times by now. But if anyone has any questions, y'all, I try to be really responsive to the comments bar and the ask a question section. So feel free, drop some questions in there, and I will definitely try to do something to explain whatever phenomenon it is you're asking about. Here we go. Let's talk about some feedback. So feedback. What do we know about feedback? Feedback is all based around this idea of homeostasis, from the Latin word homeo, which means same, to another word, stasis, which I'll be honest, I'm not 100% sure if it's Latin, but I know stasis has something to do with the word static, static meaning same. So we want everything at the same level, unchanging, same. I wish it was more creative than that, but it's like same. Point is, our bodies like to be in the same state constantly. They want the same body temperature. They want the same blood glucose level. They want the same blood pressure. They want all these things. Our bodies are needy. And we, as human beings, are so respectful of that we constantly stress our bodies out. I don't know about you, but I'm not sitting over here like eating this perfectly balanced ratio of biomolecules, getting the perfect amount of 8 hours of sleep, doing my meditation. I'm not doing all of that on a regular basis. I'm usually eating a lot of sugar, drinking a lot of caffeine, maybe exercising, maybe not exercising. And when I do exercise, I feel like my heart goes really fast. Humans live stressful lives, and part of that we put on ourselves through lots of sugar and caffeine. Part of it is just situational, though. You guys are students, and you know better than anyone else. Student lives are stressful. I remember being in high school. It's not a piece of cake. And so as a result, our bodies are constantly at this level of homeostasis that is veering and trying to balance itself out like a giant pendulum, just trying to get back in that perfect rhythm. So to describe that, I always think of this yin and yang. Everything's perfectly balanced, just swirling in order. And we are constantly trying to achieve that. But there's a lot of factors that throw us off. What's not important here is the factors that throw us off so much as the factors that restore us back to that normal. So we have this concept of negative feedback. I'm skipping over, I understand, work with me. Negative feedback. Negative feedback is this dude with the stop sign. Negative feedback is all about you're doing something that's throwing homeostasis off the rails. And we need to revert back to that norm. You're not doing it right. Stop. Go back to go. That's negative feedback. Another example I can think of is, so the idea of negative feedback is I want to go back to the normal original state. The example I often give is I have a dog. Here, I'll show her to you. Her name is Ellie. She looks like this. Isn't she cute? She had a little Christmas sweater. But so Ellie is overall a very good, very quiet, very sweet dog. But occasionally she barks. And she barks around 5 o'clock in the morning. That is like her time to jam. She barks at squirrels and cats and all these things. And as a result, me and my husband distribute negative feedback. We say, no barking, da-da-da. And through that, we restore our dog to her original normal state, which is quiet. That being said, my dog's not quiet all the time. She gets very riled up occasionally, but at 5 in the morning, the norm is to be quiet. But then on the other end of the spectrum, we have positive feedback. So I think a positive feedback is when I'm trying to get my dog Ellie to do something again. I want her to do it again. So for example, about a year ago, maybe 2 years ago, I was trying very hard to train Ellie, who is like a lab. They're supposed to be these super smart dogs, but I was trying very hard to train her to shake hands. Like whenever I would say, hold hands, she would like shake my hand. You've seen dogs do it. And so to do that, I had to give her very positive feedback that would encourage her to do that again and again and again. So for example, I would say, Ellie, hold hands. And all I would do at first was touch her paw. And she'd be like, I don't really like this. But I'd give her a treat. So she associated that with something good and she would keep doing it. I'm kind of getting into the psychology side of it. But what is really going on here is you're trying to elicit a response to happen more and more and more and more. Over time, the more treats I gave her, the more likely Ellie was to hold her paw up so I could shake it. Positive feedback. So that's like a psychological aspect to it of trying to return to the norm, which is negative feedback, versus trying to get something to happen more often, which is the positive feedback. Let's look at some actual like biology applications. What are the things that are happening in your head, in your body, that are happening in order to return your body to homeostasis? So let's take a look. So let's talk about negative feedback first. So like I was saying a minute ago, negative feedback is all about getting you back to whatever is the normal state. So for example, the most common one that we talk about is thermoregulation or human body temperature. What is the perfect body temperature of health and homeostasis that humans are supposed to be at? What is the ideal body temperature? Y'all tell me. What is our body temperature supposed to be? Tell me. I know you know. If not, Google it. Sandra, Jess, Shreya, Lucia, Madison. What is our body temperature supposed to be? In Fahrenheit, to be clear. Oh, Lucia already said in Celsius. It's okay. 37 degrees Celsius. Very good. So in Fahrenheit, that's going to be 98.6 Fahrenheit. The ideal human body temperature was previously thought to be 98.6. I say previously because very recently, like it's on my Facebook. I could pull it up now if I wanted to. One of my science teacher friends tagged me in it. They think that human body temperature is actually getting lower, which makes sense to me because every time I go to the doctor and I think I have a fever, they're like, no, you're just at 98.9. And I'm like, really? Because I feel like I'm dying. But they don't listen to me because I don't have a medical license, which is fair. But ideally, the human body temperature should be around 98.6. And so when you're at that 98.6, that's considered homeostasis, the balance, the zen, the feng shui, all the things that are right in the world. According to human anatomy textbooks, at least. So, however, we put ourselves in situations that may make our body temperature rise above that 98.6 or fall below 98.6. So what's a situation that may make your body temperature rise? What's something that happens that may make the body temperature rise? Taking a hot bath. Yes, Madison, that's a very good answer. Taking a hot bath, that's your external environment. It's on your skin. It's not on your internal temperature. But over time, if you sit in a scalding hot bath long enough, that's going to seep through and affect your internal body temperature. And as a result, if you sit in, say, a warm bath or a sauna, what does your body start doing as a result of that heat? What do you start doing when you get too hot, say in a warm bath or a sauna? You sweat. Very good, Jess. Oh, Jess, that's a good name. Very good. You start sweating, and you start sweating in order to reduce your internal body temperature and try to return to that normal, to that 98.6. This is the negative feedback. Your body is adapting in a way to make you go back to what it considers the norm, to the baseline. Now, on the flip side, let's say you are outside in a snowstorm. I don't know. I'm just thinking of things. You're outside and it's snowing, and you don't have a jacket and you're not moving around. Over time, your body temperature would lower. What does your body do in an attempt to compensate for that, to make you warm up? What do? You shiver. Very good, Madison. Very good, Sandra. You start shivering because that contraction of your muscles generates a little bit of heat, tries to warm your body up. Very good. Again, we are trying to go back to what we consider homeostasis, that thermoregulation. Very, very good. So we're going to look at another example of that negative feedback here. We're going to look at blood glucose as another example. This is one they love to talk about on the AP exam, so I really like for people to understand it. The idea is that your blood sugar, your blood glucose level rises. I often joke with my students that when I go home for lunch and eat a bowl of Cinnamon Toast Crunch, and they think I'm joking, but sometimes when I go home, I just want a bowl of Cinnamon Toast Crunch, which is, I don't know if you've ever looked at the label of most of our favorite breakfast cereals, very sugary. It's a lot of glucose. So whenever I eat those sugary foods, my blood glucose level spikes, and I get this thing called a sugar rush, which is great around noon when I've still got another class or two to teach and I need some energy so I can stay pepped and excited for my students. But here's the cascade that occurs. I eat a bunch of sugar. It's delicious. It's worth it in my head. My pancreas, which is like kind of below your lungs, is the best way I can describe its location. The pancreas receives the signal that there's high blood glucose. And as a result, it releases insulin. That insulin travels all the way through your bloodstream, all the way through. And then it goes and hits your liver, which is like right around your diaphragm, so I would say the middle of your abdomen. I'll be perfectly honest, I've never taken an anatomy course. I'm trying very hard to remember this, so work with me. But the point is, it goes from your pancreas through your bloodstream to your liver. And your liver receives those signals that, oh, hey, we got some insulin. That means there must be a lot of glucose. We got to take that stuff in. And so your liver starts absorbing all of this glucose. And as a result, your blood sugar levels go down. You return to homeostasis. Yay! That's what we want to do. Yay! Happy homeostasis. Now that's an example of hyperglycemia, high blood sugar. Now on the flip side, let's say you have hypoglycemia. Can anyone tell me what hypo means? What's hypo? Not enough. Yeah. So you'll sometimes hear of people who will say, I really need to eat some sugar. I really need to eat some candy right now. It's real. It's a thing. If a person is hypoglycemic, they don't have enough blood sugar, and that can be just as catastrophic as high blood sugar. And as a result, their pancreas doesn't release insulin. It will release glucagon. Glucagon is another hormone that will travel all through the bloodstream, woohoo, get down to the liver. But this time, instead of telling the liver, hey, you need to absorb glucose, it tells the liver to release glucose. And that glucose restores you to homeostasis. These are our examples of negative feedback because they are returning your body to homeostasis. They're trying to establish the norm once again. Do we have any questions about negative feedback? Why does it release insulin? Okay, Madison, I just saw your question about why does it release insulin. Do you mean why does the pancreas release insulin? Yes, okay. So the pancreas, like I said, it's an organ. You've got it like right below your lungs. So around here, I can't remember if it's on the left or the right. I'm sorry, you don't need to know that for an AP test anyway. The pancreas has receptors, and those receptors register how much glucose is in the blood. And so once it registers a certain level of glucose, it will either release insulin because there's too much glucose or release glucagon because there's not enough glucose. Does that make sense, Madison? If not, I have more metaphors. Okay, very good. Thank you. All right, if there's no more questions about negative feedback, I'm going to go ahead and go to positive feedback. So you'll notice I put a lot of memes and GIFs here because I think they're great. I'm going to try to move my face, though, because I'm blocked. Oh, I can't move it. No. Come back. Oh, No, that's not what I want. Wait. Y'all don't want to see. Okay, here we go. So, it's me. But I really want y'all to see these memes next to me because you'll notice they all have a common theme. What are they all talking about wanting? What do they all want, all these kids? Candy. More. Very good, Madison. All these kids want more. So you've got that kid from Riverdale who used to be on The Sweet Life. I don't know his name. I know he's a Sprouse twin. I'm sorry. But he's like, more. You've got the girl from Willy Wonka who's like, I want more. You've got the kid from Oliver Twist who says, please, sir, may I have some more? Positive feedback is all about more now. And it wants it now. In humans, there's only 2 examples that we typically discuss, one of which is disgusting, vomiting. Vomiting is a prominent example of positive feedback. If you've ever had the displeasure of experiencing vomiting, you know that once you start, it's very hard to stop. That's because there are hormones in the body that once you start vomiting, it triggers more hormones that signal to vomit. Evolutionarily, this makes sense because the idea behind vomiting is there is something that you have ingested that is now in your system that you want to eject from your system. So the act of vomiting is to make sure you get all of it out of your system. I apologize for anyone who's eating dinner while watching this. But on the flip side, another example of positive feedback, that more, the most common one is childbirth, because when a child is born, it sits in this little sac. I do know where this one is. It's like right below your belly button. And that little sac is called a uterus. And the uterus is a muscle as well. It's this little sac. But when you put a baby in it, it gets bigger because a small football-sized object has to fit inside. And so this baby, when triggered by a hormone called oxytocin is considered a love hormone. It is triggered when you have muscle contractions. Sometimes those muscle contractions are as simple as a hug, the muscles in your arms of an embrace. But typically, oxytocin is the contractions of the muscle of the uterus, which is literally squeezing a baby out of a human being. And so this is positive feedback because one contraction triggers more contractions. Think of it this way. If you've ever heard of anyone in labor, they always say you need to count how long occurs between contractions. At first, it'll be like 10 minutes between every contraction. Then it'll be 5 minutes between every one. Then it'll be 1 minute between every one. So the longer it goes on, the more it occurs, more. That's our positive feedback. Another example of positive feedback, not in humans, in fruit, would be ethylene gas. Plants, fruit specifically, release ethylene gas. It's a method of autocrine signaling. And so they signal themselves, oh, hey, time to get ripe. So when you're out there shopping at the grocery store, looking for some good bananas or avocados or whatever it is, you want your fruit to have experienced a little bit of ethylene, because that ethylene causes the fruit to ripen. The problem is if your fruit is next to another fruit, it will cause other plants to ripen, because sometimes that ethylene gas floats over, triggers the next fruit, and it's like, whoop, whoop, time to get ripe. So it can cause, so to speak, one bad apple or one ripe apple spoils the bunch. One triggers more. Positive feedback is all about more. Negative feedback is all about go back to homeostasis, go back to the start. Any questions about positive feedback? Going in 3, 2, 1. All right, we're going on to the cell cycle, so I hope you guys are ready. A doom. Do you guys watch TikTok? Because lately, I just really want to make a bunch of biology TikToks. I keep getting the song stuck in my head. And I think of that one that it's like art, where the girl is drawing something. And that's what I think of when I see this particular GIF. You see it starting as in, here it goes, wait, an infant. And the infant becomes a child, and the child becomes an adult, and the adult becomes an older person, and the older person becomes a ghost, and it goes back to being a child. So in the cell cycle, we have a similar life cycle. We as humans go through that life cycle of child to adult to older person, so on and so on. Cells go through the same thing. They have a life cycle. They have a pattern that they follow. That pattern is illustrated right here. So we can see the actual physical what occurs. You've got this red dot, that's a cell. And it, oh, sorry. Whoa, we're not ready. There we go. And it almost looks like it's struck by lightning. And then it slowly pinches apart. What do we call that process where the actual cell splits apart? What is that process called? Mitosis. Very good, Madison. So in mitosis, the cell splits. But mitosis is one short part of the cell cycle, much like reproduction for humans or having children for humans. It's just one part of our life cycle. We have a bunch of other events. So in the cell cycle, our other events would be G1, S, and G2. So G typically stands for gap or growth, as in gap 1 or growth 1. S is for synthesis or DNA replication. DNA must repeat itself or replicate itself before you can divide a cell in half. Otherwise, you wouldn't have enough DNA in each cell. G2, gap 2, growth 2. The cell has to grow a little bit more. It's got to get ready. Mitosis, we've already talked about, is going to be this phase right here where the cell actually divides and creates those identical daughter cells. And then we have this G0. I like to think of G0 as, why do I keep doing that? I'm sorry. G0 as this go. G0 means go as in get out. Because what happens when cells are put in G0? What does that mean? Y'all tell me. They stop growing or replicating. Very good, Shreya. So G0, I like to think of it as cell retirement. It means the cells are not growing, they are not dividing, they have been put out to pasture, so to speak. They have no more business involved in growth or development. They've done their dues. They don't need to do any more. G0 is very important because without it, we would have cells that are replicating and trying to grow, even if they may be unhealthy or if they may have improper division. I see Madison asked, do cancer cells have the incapacity to go to G0? Yes. So G0 is what cells are supposed to do. After so long, a cell doesn't need to divide anymore. It doesn't need to grow anymore. It just needs to be done. It's done its job. But sometimes cells miss the checkpoints, which we're going to talk about in a moment. And as a result, they skip G0 and they divide anyway. And that's how cancer cells develop. So Madison, very good question. Without further ado, I'd like to go ahead and go into that. Does anyone have any questions about the cell cycle, though? Okay, quick tip, to my knowledge, which is, I like to think pretty good, you will not be asked any questions about the specific steps of mitosis or meiosis on the AP Bio exam. They may ask you things generally about how the cell divides with the spindle fibers and the metaphase plate, but they won't ask you, like, describe prophase, metaphase, anaphase, telophase. They won't do that. It says so in the book. So I'm not going to go into super detail about that. I'd rather focus on the regulation part because that's more of the application. If you need to memorize the steps of mitosis, there's this thing called Google, and you can type in steps of mitosis and then make yourself some flashcards. So I'm going to go ahead and go on. But if anyone has any questions at the end and we've got some time, I'd be happy to field those. So let's take a look here. Speaking of that mitosis, it goes around in a similar cycle, except we see these new words here, interphase and cytokinesis. What does cytokinesis mean? What is cytokinesis? Division of the cytoplasm. Very good, Shreya. So cytokinesis is going to be when the cell actually chops into daughter cells. It splits in half and separates all the way. What is this other one? Interphase, though. What's interphase? What is interphase? Tell me. In between division. Very good, Madison. Interphase is going to be every single stage except for mitosis. Growth, replicating DNA, making more organelles. Very good, Shreya. Interphase is everything except mitosis. So G1, S, G2, G0. It's where the cell is just hanging out, dividing, doing all the things it would normally do when it's not reproducing itself. And so the majority of time is actually spent in interphase. 90% of a cell's life is spent in interphase. They only spend about 10% of their time in that mitosis, in that splitting. Even though it's so many steps to go through prophase, metaphase, anaphase, and telophase, that's only 10% of a cell's life. So it goes by pretty quickly for them. Now, cells can divide in a couple different ways. Like I said, you don't need to memorize these, but I want to go over these illustrations really quickly. We're talking about mitosis, where they're going to go through this first splitting, and then they're going to make more DNA in the S phase, synthesis, because if you don't have enough DNA, you can't have complete cells. So you've got to have that DNA replication. But after that, the cells are actually going to divide. You can see here, metaphase, they're all lined up along the middle of the plate. Anaphase has already occurred here. We're in telophase. That cleavage furrow starts to form. And by the end, we've got complete cytokinesis, where the cell has completely split. Meiosis, basically the exact same thing. It just happens twice and with more genetic variability because we're forming haploid cells instead of diploid. Now, we've got this thing called binary fission over here. Just out of curiosity, can anyone tell me what binary fission is or who uses binary fission? Bacteria. Very good, Tria. Prokaryotes, particularly bacteria, are going to be using that binary fission. Binary fission is different in that it is basically cloning. They create pretty identical cells. There can be some mutations, but generally speaking, fission is going to result in completely identical cells. Okay. So let's talk about the regulation. So I think of cell cycle checkpoints as kind of like Fred here. We've all probably been through these things at one point in our lives or another. They're called turnstiles, where you have to push the little gate in order to pass through the gate. It's called a turnstile. I like to think of the cell cycle as having 3 turnstiles that you must pass before you can get through the whole cycle. So those turnstiles are going to be at G1, G2, and M. So at the start of growth 1, at the end of growth 1, at the end of G2, and in the middle of the M phase. All of these are going to be dedicated to making sure that the cell divides properly, that there are no mutations, that the DNA divided properly, and that there's no cancer. So, for example, in the M phase, the M checkpoint is concerned with making sure that the cells, when they divide, all the chromosomes were equally split onto opposite ends of the new cell. What is the M checkpoint concerned with preventing? It's all about making sure you have the right number of chromosomes on each side. What is the M checkpoint concerned with? So it's anaphase because it's splitting. I guess I should be more specific here. What kind of disorders can arise if they skip the M checkpoint? If the cell's chromosomes aren't divided properly. Down syndrome. Very good, Tria. So these are going to be your chromosomal disorders. If you don't do this properly, too many chromosomes. Very good, Madison. You can have too many, too little. Ideally, we want a karyotype that looks like this one. Oh, it's doing that annoying thing again. Hang on, let me see if I can. Sorry, my face is big, but at least now you can see the chromosomes. So you can see here, they're all in pairs like they're supposed to be. Now, if you had one that was just one chromosome, or had 3 chromosomes, that'd be bad. Excuse me. I would edit that out, but this is a live stream. Sorry, y'all. So we want to make sure we have the right number of chromosomes. And here in purple, this little illustration is meant to demonstrate cancer cells because they often are uncontrolled cell growth, which means they grow very fast. And as a result of growing so quickly, they don't always do it properly. Now, for this cell cycle to happen properly, we have these checkpoints. It's kind of like you going through a metal detector. It makes sure you're not bringing anything metal in. Ignore this guy who is just bypassing. This is our little cancer cell who is skipping through the checkpoint even though he's not supposed to. So we've got a checkpoint in G1, G2, and M. G1 is just going to be about making sure that the cell is growing, dividing, it's got enough materials, it's healthy. If it's not, or if there's a problem with the DNA, where is the G1 checkpoint going to send the cell if it can't go on to the S phase? G0. Very good, Tria. Very good, Madison. If the cell is not doing what it's supposed to, if it's not healthy, or if it's just old, the G1 checkpoint may say, you know what? You don't pass the test. I'm sending you to G0. Happy retirement. And that's okay because we don't want our cells going on if they're not healthy. Then in the S phase, we've got to have our DNA replicate. If there's an error in DNA replication, if there's a mutation, what is the first checkpoint that should catch that error in DNA replication? Who should catch that error? G2. Very good, Tria. So we see DNA synthesis. That S phase is going to occur right here. If something goes wrong in here, it won't be caught until G2, and it is critical that G2 catches that and make sure that the cell is big enough, that the DNA looks right, that everything is gone on without a mutation. If G2 doesn't catch that and the cell goes on to mitosis to divide, that's where you can lead to cancer. The M phase, again, it's just making sure, hey, did everything divide properly? Do we have the right number of stuff? If not, that can also lead to cancer or a chromosomal abnormality. Does anyone have any questions about those checkpoints? Three, two, one. All right, I'm going to go on. You guys are doing great. All right, now here's the part that's new. We have these things called cyclins. If your teachers use Pogles, which are those like worksheet books that they have you look at the picture and find the answers, you may have already done something like this. Cyclins and cyclin-dependent kinase. So in order for a cell to divide, you need a certain level of hormone to happen. So the way I think of this is, if I want to knock down a wall, let's say there's a wall in my house. Let's say it's this one behind me. I don't like it. I don't like this wall. I want to knock it down. To knock down a wall, I would need a certain amount of strength in order to physically crush the wall. Okay? Madison, I'll come back to your question in just a sec. So to physically crush that wall, I need enough strength. In order for a cell to divide, it needs a certain amount of a hormone or a signal. That signal is the cyclin. So we can see in these graphs here, there are a bunch of different cyclins, D, E, A, and B. And you have different concentrations at different levels. You use cyclin D at G1, cyclin E in the S phase, cyclin A in the G2, and cyclin B in the mitosis phase. These cyclins bind to something called CDK, cyclin-dependent kinase. What is kinase? What is kinase? It's an enzyme. Very good, Shreya. Here's how I describe kinase to my students. Kinase is a light switch. It goes off and on, off and on. It turns on whenever I add a phosphate to it. So it turns off whenever I remove that phosphate. Kinase is the light switch, flick, flock, flip, 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 that turns reactions on and off in your cells by adding a phosphate or removing a phosphate. You can actually see it here where the phosphate is bound or phosphorylated. So when cyclin binds to when cyclin binds to kinase, that cyclin-dependent kinase, and that CDK cyclin complex is phosphorylated, it turns on. When enough of these molecules turn on, it triggers each step, whether it's G1, S, G2, or M. They have to reach a certain level in order to turn on the reaction. Okay, coming back to your question, Madison, do the cells have to miss both checkpoints or just one to become cancerous? So it really depends. I'll be perfectly honest. I like to think that if a cell skipped G1, somehow it became cancerous in G1. And all cancer means is unregulated cell growth. So by that, we can go by a lot of things. It's growing too fast, it's growing too slow, it's not growing everything, it's growing too much. Whatever that is, let's say it's happening in G1. I find it highly unlikely it wouldn't be detected after all of these checkpoints. But each checkpoint only checks for certain things. It'd be kind of like if we were working in a car factory. I would check tires. You would check windshield wipers. You would check chairs. I only check tires, so I'm not going to check and make sure that the airbags work. So if something skips one checkpoint, excuse me, my allergies are getting me. If something skips one checkpoint, Madison, it's not like it's skipping the second checkpoint. The second checkpoint doesn't check for it because that's not its job. Does that make sense? Okay, very good. All right. Oh, I see another thing. Yes, cyclin CDKs are part of the cell cycle checkpoints. So the way I have been teaching it, cyclin CDKs are not the checkpoints. Cyclin CDKs are what activate it to go on to the next part of the cycle. So when you get enough cyclin D, that triggers it to go on to S. When you get enough cyclin E, that triggers it to go on to this, and so on and so on. They're not necessarily the checkpoints. The checkpoints are going to be related to different things. These cyclins, though, are going to make sure that the cell cycle is going on as expected. The checkpoints are to stop it when something is going wrong. So I guess you could say the cyclins are like the positive feedback. It tells it more and more and more, keep going, keep going, keep going. And the checkpoints are like the negative feedback. They try to return it back to normal if something has gone wrong. Does that make sense, Shreya? Coolio. Okay. So, quick update here. Guys, you will see all sorts of things if you try to Google this. Talking about cyclin with a K and cyclin D, E, da, 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 da. Don't worry about that. You don't need this for the AP exam. Let me make my face bigger to emphasize this. You don't need all the big words with all the hyphens in them. They're not relevant to you. They're not relevant to the AP exam. If you were in medical school or getting a PhD in medicine, yeah, you'd probably need to know that. But for right now, all you need to know is that cyclins and kinase turn the process on and off. Excuse me. I think I'm talking too much. Now, when those checkpoints fail, we do get uncontrolled cell growth. And that can result in a cancer. That can result in a DNA problem. That can result in apoptosis. The cell may get sent to G0 and it may be like, actually, can we just like delete that cell? And that's apoptosis. But if you look here, it can also lead to other abnormalities. If you look at this karyotype, what's the abnormality here? What's the abnormality on that karyotype? 321 chromosomes. Very good, Shreya. That's Down syndrome, trisomy 21. So in this case, something went wrong at the M checkpoint, the metaphase checkpoint. The chromosomes weren't divided properly, and so too many went over on one side. That's how you end up with something like Down syndrome. Very good. Madison, what is kinase again? Kinase is an on-off switch. So it's a molecule. Let me draw it for you. So let's say this is kinase. Pretend this square is kinase, okay? Now, when I add a phosphate, I know that looks like a Q because it's backwards. Just roll with it. When I add a phosphate, it turns this kinase on. And when it's on, it triggers another process to occur, whether that's move on to the G2 phase or send out a neural impulse, whatever it is. That phosphate turns it on. It makes it do a job. It's the same as when I turn on a light switch. Now, if I take that same kinase and I dephosphorylate it, that is, I take off the phosphate, now the kinase goes from on to off. It's just an on-off switch. Does that make sense, Madison? Cool. Glad to help. All right, y'all. We're a little early, but that's my last slide. That's all I got. Do y'all have any questions for me? What can I help you with? What are your burning questions? Secrets of life? Things like that? Questions by my cactus? I painted that myself, just so you know. What questions do you have? The insulin pancreas process, yes. So, gosh, I do not know where this cough is coming from. I'm so sorry. Okay. I'm going to back this up a little so you can see me. Because I'm going with illustrative examples here. Okay. To review, you've got a pancreas somewhere in here. I don't know if it's on the left or on the right, but you got a pancreas somewhere like under your rib cage, underneath your lungs, behind your rib cage, underneath your lungs. You have sugar, glucose. So, Madison, let's say you had, I don't know, cake for dinner. Let's pretend you had cake for dinner. There's a lot of glucose in cake. That's why it's so delicious. Your pancreas is constantly reading the blood sugar level in your blood. So it's exactly the same as someone with diabetes who has to test their blood sugar with that meter, except your pancreas is doing it all the time because it has receptors that say, how much blood sugar is this? How much blood sugar is this? When that blood sugar gets too high, your pancreas basically sends off a fire alarm that says, nope, 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 this is too far from homeostasis. This is too far from where we're supposed to be. And as a result, your pancreas releases these things called insulin. They're big steroids. And that insulin goes all the way through your bloodstream and all over your body. But your liver has receptors. Your liver is like around your belly button, somewhere in here. And your liver has receptors for insulin. And so it picks up that insulin. And when the liver receives a signal from insulin, it tells the liver, oh, hey, time to absorb glucose. It doesn't know why. It doesn't care. It just does as it's told. And the liver absorbs the glucose. And as a result, your blood sugar level returns to normal. Does that make sense, Madison? If not, let me know. Okay, cool. Yay. What other questions do y'all have? Y'all got me for 11 more minutes. Oh my goodness, girl. Madison, that's so much work. Okay, well, I'm here like semi-often. If you ever need more help, here's my Twitter handle. Feel free to just like tweet questions at me because it gives me life. Literally, my Twitter is just all the random things I think of to do with my students. So happy to help if you ever have questions because that's a big task. But you're doing good. So good job. Anyone else have any other questions about feedback or mechanisms? Or cell cycle regulation? All right, y'all. I don't hear any more questions. It is 7:50 my time, and I'll be perfectly honest, I'm an early riser. Oh, wait, no. Can you help me with something not about the cell cycle? It depends if I know it. Just go ahead and ask, and I'll see if I can help you. Anything for another Jessica. Ask your question. Ask away. Also, I'm sorry I'm blinking so much. I've had contacts in since like 5 this morning. Is this for homework? Just out of curiosity. Very good, Madison. Small nonpolar molecules. Small and not charged. Very good, Sandra. Hey, totally understand. It just sounded like a question that came out of one of my labs, so that's why I was like, that sounds familiar. So Jess, you have a cell membrane made of phospholipids. Lipids, if you remember, are non-polar. Non-polar stuff doesn't mix with polar. Like mixes with like. So if you've got molecules that want to pass through that cell membrane, you need small non-polar molecules because they must match. Polar things will have a charge and they won't match. But hey, Madison, Sandra, air high five. Great job, y'all. Whoop, whoop. Y'all know science. All right. What other questions do we have? So just water, some polar stuff can pass through, but only like 1% of stuff will pass through. Like if you had like, say, a milliliter of water that needed to pass through a cell membrane, you would get like 0.01 of that milliliter through. Water typically is actually going to move through cell membranes through a special protein, a special channel protein called aquaporins. Pretty much anything polar is going to need a protein to get it through. Make sense? All good. Whoop, whoop. You're welcome. Just in case anyone's wondering, I will be back next week. I'm trying to remember, what am I teaching y'all next week? I'm going to stop my share because I'm about to like go into my Google Slides to try to remember what I'm doing. This is a little out of order from how I teach it. So, and in case you're wondering, yes, this is exactly how I act in front of my students. So, whoop, whoop. It looks like I am teaching meiosis and an introduction to heredity next Wednesday. So if you have questions about that, I'll be here this time next week at 7 p.m. Central to talk about meiosis. So that'll be expanding on the cell cycle a little bit because meiosis is like the extra version of the cell cycle, if you will. This is my star wand. For when you do a good job, you get a gold star. By the way, you all get gold stars. Thank you for being here on a Wednesday night. What other questions do we have? You've got me for another 6 minutes. Three, 2, 1. All right, y'all. I'm going to make the executive decision then and say, time's up. Y'all have been an awesome crowd. Thank you. Thank you. If you have any questions, seriously, that's my Twitter handle. Feel free to message me. I can't guarantee I'll respond to it because notifications are turned off on my phone because I try to not let social media run my life. But if I see it, I'm so happy to help. So I hope I see you guys here next week. Thank you for coming. Sandra, Shreya, great to see y'all again. Jess and Madison, very nice to meet y'all. Hope y'all have a wonderful week, and hopefully I'll see y'all next Wednesday. Bye-bye.